in the right spots. Okay. All right. Okay, so thank you so much for being here. My name is Maura Niemisto. I am a research associate at Bigelow Laboratory of Ocean Sciences, which is in East Boothbay, Maine. I want to thank Alicia and the Carpenter's Boat Shop for inviting me to talk today, and thank all of you for tuning in on a Thursday night. And today I'm going to talk to you about the work that I've been participating in the last 7 years or so, which is this question about larval lobsters and why their numbers are declining. Now, my goal today is to give you a summary on as many pieces of the puzzle as I can that we've been working on. And I realized that as I was putting this talk together, it would take me a week to give you an in-depth summary of all these projects. So it's probably going to be a little more quantity over quality, and you can, I'll give you some resources if you're interested in looking in depth at any of the subjects that I talk about. I'm going to highlight many projects, and you'll see names of many researchers, graduate students, and interns during this presentation. And there have been just so many contributors and funders and great minds working on these questions. I don't have time to introduce all of them, but I can talk a little bit about me. And since this is a Carpenter's Boat Shop presentation, I'll mention this little piece of my history, which is what actually brought me to Maine and marine science, which was sailing in wooden boats. In 2013, I actually moved out to the East Coast. I'm from the Midwest. I moved out to the Hudson River Valley, and I lived and worked on a 108-foot traditional Hudson River sloop called the Clearwater. And if you've never heard about it, this is a project that was started in the late 60s by singer and activist Pete Seeger with the mission to bring students and folks living along the river out to the water to foster a sense of ownership and interest in taking care of their watersheds, as well as to teach about estuary ecology. Incidentally, this is also where I met Chelsea Fisher, who is the program director over at the Carpenter's Boat Shop. So I was kind of connected even before I moved to Maine. But this is also in this role as an onboard educator is where my interest in marine science kind of took off. And I decided to go back to school to get a master's in marine science and to build a better foundation as a marine science educator. So I started my work, my master's thesis work on larval lobster and climate change in January 2017. And I was co-advised by Dr. David Fields, who is a research scientist at Bigelow Laboratory, as well as Dr. Rick Wally, who is, well, he just retired actually from the University of Maine Starling Marine Center, as well as the director of UMaine's Lobster Institute. And he was a previous board member at the boat shop. So he's a local guy. If you're local, you probably know him. But after graduation, I was hired on to work as a research associate in David Fields' lab, and have continued to work with both Rick and David on a number of lobster projects over the past 5 years. Now, incidentally, lobster is actually just a small piece of the pie of everything I work on in the Fields lab. We are a zooplankton physiology and sensory ecology lab. So we study a breadth of subjects pertaining to many, many species. If you're fuzzy on the definition, zooplankton is a group that is made up of animals. That's the zoo or the zoo part of zooplankton, who are up in the water column and moving around by current. So that's the plankton part. Planktos is Greek for drifter. So zooplankton are animal drifters. And we've worked on a ton of different questions about a ton of different zooplankton species, ranging from like the effects of oil spills on Calanus copepod populations to alternative methods of mitigating salmon lice infections, which are a copepod, to effects of seismic blasting on zooplankton populations, just to name a few of the different projects. And these are, this is just a smattering of different animals that we've worked with over the past few years. And there are even more. Here are some more animals that I've worked with. This one takes a minute to load for whatever reason. Oh, there we go. So our lab is also very connected to the education programs run through Bigelow. I'd be remiss not to mention them here in case anyone knows of some young folks who are looking for hands-on lab experience or are interested in marine sciences. We have a week-long intensive program for high school students in the spring. It's called Keller Bloom. We have a summer internship programs through the National Science Foundation's research experience for undergraduates. We usually have about 30 students living at the lab all summer long. And of course, we run a semester program affiliated through Colby College, although you don't have to be a Colby student to do it, called Sea Change Semester. That's actually happening right now at Bigelow. And in my role, I help facilitate the research cruises for all of these courses, as well as some of the lab work that they do. All right, so, but y'all came here to hear about lobsters, so I'm going to introduce the star of the show now. So when I say lobsters, is it going to go? Perhaps this is what you think about. You might think of a lobster dinner or the festivals and parades and celebration of lobster as a cultural icon here in Maine. Or maybe you think about the working waterfront and the incredibly resilient, unique fishery we have here in Maine. Or maybe you just think about traffic across the Wiscasset Bridge as tourists run out to grab a picture in front of Red's lobster roll. So it's an extremely important icon, and it's intricately tied into the socioeconomic and cultural framework for the state of Maine, which I probably don't need to tell any of you. Maybe when I mention lobsters, you even think about this. This is the star behind the most valuable single species fishery in North America. That's a American lobster on the seafloor. This is the value, this pie chart is depicting the value of the commercial landings in Maine alone from 2022, totaling at over $500 million. Now, nearly 70% of all that value came from lobsters. So we care a tremendous amount about these animals and how they might be responding to changes in their environment. Luckily, adult lobsters are a model organism for a lot of biological research. They've been studied for a very long time, and lots of funding and effort is put towards expanding our knowledge and understanding of them. Now, for me personally, when someone mentions lobster, now this is all I think about. This is a newly hatched stage 1 larval American lobster, like right out of the egg. It's about the size of a peppercorn. It lives up in the water column, feeding on smaller organisms, and it's moved around by the current. So that technically makes this a zooplankton, which boots it into my area of expertise as a zooplankton biologist. But unlike the adults that we have studied for a very long time, we actually know relatively little about larvae. So there's a lot of good questions and avenues of study that have not yet been explored. All right, so to set the scene for this talk, I'm going to walk you through a quick life history lesson for lobster. All right, an American lobster starts its life out as an egg stuck to the abdomen of an adult female. At the bottom, they hatch when the water reaches a critical temperature in the springtime, and they emerge as a stage 1 larvae that drifts up into the water column. At this point, they're small, kind of insect looking, and they're fairly poor swimmers at this stage, but they're very cute. They go through a series of molts through several larval stages, stage 2 and 3. Each of these stages is a little longer in duration than the last. And each time they have to shed their exoskeletons and molt to become a little bigger of a stage than the last. After this point, they go through a major physiological and morphological metamorphosis into a post-larval stage 4. This is what we call the Superman stage. And during this stage, the animal starts to look like a lobster, but it is still up in the water. You can see its pose of its arm is where it gets that Superman stage as it swims through the water column. They're really good swimmers and really good hunters, and they're moving up and down looking for really good bottom habitat to settle into and molt into a benthic juvenile, or a juvenile that sits at the bottom. So in optimum temperatures, this whole process takes just over a month to complete, and it will take around 7 more years for that lobster to grow into a harvestable size of something that you would purchase to cook. So during these early stages, larvae are subjected to the dangers of the water column. So up in the water column, temperatures can fluctuate more quickly than at the stable bottom, and they're much more vulnerable to predators swimming around. And it's within this context that they have to find food and survive through each of these stages. Now, surviving through all the way to the post-larval stage is an incredible feat, and very few of these thousands of larvae hatched from each clutch will make it through. These transitions between stages are important biological bottlenecks, meaning that they're places where fewer and fewer larvae survive to each consecutive stage. And one thing that we're trying to understand is how stressors such as changing water conditions, food availability, etc., might alter or restrict these bottlenecks even further. Understanding what these stressors are and how they're affecting the larvae help us predict population dynamics down the line. All right, so it's within this context that I'm presenting the question that's driving all the research that I'll be highlighting. And it's a paradox that Rick Wally has coined the great disconnect, which I'm gonna call today, what's happening to the baby lobsters? So over the past 50 years, lobsters have already kind of demonstrated this northward population shift, which has been attributed to warming. So this is a GIF that shows relative lobster abundance within the US range from the late 60s to 2014. As temperatures have warmed, these populations have moved northward and into deeper waters. Now, as more lobsters have moved into the Gulf, we've seen a huge upswing in the commercial fishery here. Since the early 2000s, landings have doubled, and we've seen more and more eggers, that's females with eggs, and as a result, an increase in the number of stage 1 larvae. However, and here's the disconnect, even though stage 1 larvae have continued to increase in numbers, since 2012, we've seen a precipitous decline in the number of post larvae. And resultingly, that has resulted in a decline in the number of juvenile recruits that are making it to the bottom, where they'll grow up to adult size. There's a great paper down here that talks about that. Okay, so this begs the question, what's happening to the baby lobsters? In other words, what stressors are narrowing that bottleneck and causing fewer larvae to make it to that stage 4 than we might expect? And with any question in biology, especially in the ocean, which is such a complex system, the answer is likely nuanced, complicated, and of many parts. And we've been working on better understanding this, poking at it from several different directions. So a couple that we've looked at have been climate change, larval quality, food availability or food limitation. And I'm about to attempt in my remaining time here to highlight a handful of our projects that have poked at these different questions. And I'll start with a project that was tied into my master's work, since I know the most about it, which is looking at the joint effects of ocean warming and acidification on larval lobster performance and gene expression. So listed at the bottom of the slide here, you'll see an army of people who are part of the beast that was this study, including a half dozen RU summer interns. So the Gulf is warming. Here's a map of the world's oceans depicting the change in average sea surface temperature over a 10-year period following 2004. Red is an increase, blue is decreasing. The Gulf of Maine, which is pointed out right here, is bright red. It is experiencing an alarmingly rapid change in temperature, and it's been noted in several studies as one of the fastest warming regions in the world. Now, most marine organisms are ectotherms, meaning cold-blooded, and their metabolic systems and basic biological functions are really closely tied to temperatures. So changes in temperature have incredible potential to impact our oceanic ecosystems. So as the Gulf is warming, it's also experiencing acidification. This is, I'll cover this really briefly. It's the process in which carbon dioxide is absorbed into the ocean, which is altering the chemical properties of the water, lowering the pH, and making it more acidic. So one result of this is that there's less available material for organisms to build and maintain their shells. It's a special concern for animals like clams and oysters and crustaceans, all things that maintain calcium carbonate shells. Now, the Gulf of Maine is, of course, particularly susceptible to this phenomenon. One factor that can increase the risk is freshwater input. This is a map of all the freshwater bodies of water feeding into the Gulf. And that glut of fresh water entering can lower the buffering capacity of the Gulf. That's which in a sense is lowering the ocean's natural defenses to protect against that pH shift. This can result in faster acidification. All right, so double whammy here in the Gulf. What about lobsters? We talked a little bit about warming already, but in terms of acidification, the adults are known to be pretty resistant. The bottom tends to be a little more stable, and adults actually have the ability to manipulate the pH in their body, so they have some defenses there. However, we don't really know as much about larval resistance, and they're seemingly more vulnerable. For one, they're living in the water column, which tends to experience quicker fluctuations in temperature and pH, and they're going through these really demanding molts through each of these stages in fast succession, all the while spending energy looking for food and avoiding predators. So each of these stressors has a potential negative impact on the lobster, but warming and acidification are interconnected and happening simultaneously. And what is becoming clear is that when we ask questions about how individual species will be affected by a changing ocean, we need to look at how these stressors act in combination, since multiple stressors can have combined negative effects greater than either stressor alone, resulting in an animal that can be very stressed out. All right, so this study sought to understand the response of larval lobster to oceanic warming and acidification, both as independent stressors and then together as combined or joint stressors. And we decided we were going to ask this question by raising larvae from stage 1, freshly hatched, through all the way to their post-larval stage in these different tanks simulating these stressors. So we raised some in ambient water, which is 16 degrees Celsius at that time was the sea surface temperature average in the summer. In some tanks, we warmed 3 degrees. In some tanks, we acidified the water. And in some tanks, we both acidified and warmed the water as a joint stressor treatment. So we raised thousands of larvae in these treatments and measured an array of different metrics along the way. We looked at growth, how big they were getting, development time, so how long it took them to get each stage, their oxygen consumption, which tells us about their metabolic rate, and even their weight, and some behavior metrics as well, such as swimming speed and feeding behavior. All right, and the big takeaway that we found in these studies was that temperature repeatedly showed a stronger effect on larval performance. So in warmer temperatures, larvae moved through these stages faster, but they were smaller and had a higher metabolic rate and a higher mortality. And though acidification effects were detected in a few of the endpoints, temperature really dominated as where we were seeing the greatest effect. So this result aligns pretty closely with what other studies on larval lobsters have found when looking at joint stressor questions. And so we said, great, if we take the results of this study and others like it at face value, we might be able to make a statement that larvae appear to be pretty resistant to acidification, and that warming is in fact much more of a concern for these animals going into the end of the century. But as scientists, and you might know this about us, maybe some of you are scientists, we're very seldom satisfied with our first answers. So we thought that this result begged the question, really? Like, are these tiny crustaceans actually really resistant to acidification, or are we just looking in the wrong places? So that led us to this project of looking for hidden responses on the molecular level that maybe we weren't able to see in our experiments on our whole body physiological experiments. So we have another tool to look at this question, and that is high throughput genetic sequencing. What this tool allows us to do is pull RNA from our animals and look at differences in how they're up and down regulating different genes when exposed to different stressors. So I have a little microbiology review here as a preface for how this works. In our cells, or in a lobster's cell, we have DNA that codes for all the genes we have and could potentially utilize. When an environmental signal occurs, or something in your cell needs to happen, a signal is sent to transcribe that a gene from that DNA into RNA. That RNA will, the RNA will carry the code out of the nucleus, and that will be translated into a protein. In this case, that gene was upregulated. Another way we could think about this is that DNA tells us all the functions that a cell could possibly do, and RNA tells us what a cell is actually doing within that moment. So what we use this tool to ask the same question of our reared lobsters. How do ocean warming and acidification affect host larvae specifically as singular and joint stressors? And this is kind of what a result of this technique can look like. This is a heat map. It's showing just one of our treatments. All of these green and red and black lines stacked on top of each other represent those over 1,100 genes that we found were differentially expressed in our 4 treatments. The green represents an upregulated gene, and the red indicates a downregulated gene. So let's look across treatments. When compared to the control, this is temperature. This is in our warmed treatment. We found that 200 genes were differentially expressed, which is a lot. But then look at this acidification treatment. In this treatment, we saw almost 500 genes differentially expressed, which is more than double that of temperature stress. Finally, in the joint stressor treatment, we saw an even stronger response. It lit up like it was Christmas, with 900 different genes switched up or down, more so than either stressor alone. They both combined to be even more than that. I'm going to stop here just to remind you that previously these types of studies had always indicated temperature to be a stronger driver of response. But on this level, when we looked inside the cells, we saw a really different story. So we were excited about this finding. It's not so much good for the news for the lobsters, but we were able to see something no one had ever seen before. And these effects didn't show up in our whole body physiology studies, which indicates that all this activity going in their cells might be hints that they have tools to compensate for some of these stressors and that they're actively doing so. But these processes of building proteins are energetically expensive, and that energy has to come from somewhere. Some of that energy can come from eating more. That's one hypothesis. Like when you hike up Mount Katahdin, you need to eat more trail mix to compensate for all the energy you're burning. And one thing about our previous study is that we fed our lobster as much big, slow brine shrimp as they could possibly eat. They were never food limited. And that might not be realistic in the real world, because in the ocean, food is way more patchy and often not as easy to catch. So of course, in true scientific fashion, these findings just made us want to dig deeper And we found ourselves asking, in a food-limited ocean, would we be able to detect some of these effects of acidification? And that led us to a new angle to investigate in a project that I call Hunger Games, in which we looked at the effects of elevated temperature and acidification on the metabolics and lipid stores of starved American lobster larvae. This experiment was actually done in 2018 by an RU intern named Adrian Contreras, an undergraduate from Palomar College. So he ran the experiments, and we have since analyzed his samples and interpreted the data. This study solely focused on the very first larval stage because they had never eaten before. The success of this early stage to make it onto the next stage has been showed to be linked with the availability of essential fatty acids and lipid nutrition that a larva can be supplied with. So very few larvae will molt to the next stage if they can't find enough food. All right, I'm going to lay this out. So under good conditions, when hatched, larvae will be endowed from its mother with a yolk sac, pictured here. These are critical lipid resources that can provide energy and carry the basic metabolic processes that this animal needs until it can find its first prey item. In this example, let's use a copepod to represent its first feed. Of course, finding food can be a matter of luck, since I mentioned the ocean is patchy. So we can imagine that from the moment this larva hatches, it is introduced to a ticking time clock in which it has to find food in order to start building its own energetic reserves to be able to molt to the next stage. So as our larva is searching for its first feed, it is actively depleting that maternal lipid store. It's running out of time. Now, how long that larva has to find its food is based on how fast that store is depleting. And this is dictated by yolk quality. That's like how much the mom gives it in the first place. Metabolic rate, so how fast it's burning through that reserve, and also food availability. So we can also imagine that in an environment where this larva's metabolic rate is being impacted by environmental stressors such as warming and acidification, it will have less support from its maternal stores and a faster burn rate. And as a result, it'll have less time to find external energetic sources like food, a higher risk of starvation, and a good possibility of fewer larvae reaching the next larval stage, which is a restriction of that bottleneck. So for this study, we had the same treatments as before. Oh, this is our study question. How does ocean warming and acidification affect the starvation resistance of newly hatched lobster larvae? So we had the same treatments as before, but now we had to keep each larva in its own individual jar because unfortunately they tend to eat each other if you put them together, and that definitely affects their lipid stores. This experiment lasted for 6 days. On day 2, 4, and 6, we pulled larvae from all treatments to analyze their weight loss. So I'm going to show you a graph here. This shows the rate of loss of a group called fatty acid methyl esters. It's just critical fats that they need. This is the group of fats that a mother would endow her offspring with. This includes like omega-3s and 6s and all those good things. So on the x-axis, we have the experimental day, and on the y-axis, we have the percent of those fatty acids left by that given day. These are all the treatments pooled together because I just want to introduce you to this concept here. So in the first 2 days, larvae lost 30% of their fatty acids through starvation, and that is the most effective weight loss program I've ever heard of. It appears to be the time where the most critical loss happens within this experiment. By day 6, they had lost 40%. And at this stage, they're entering a catatonic state, meaning that they have lost their opportunity to molt to the next stage. Now I'm going to show you how acidification and warming affected these rates. We have the dashed lines represent acidification. The redders are the temperature, and the red dashed line is that joint treatment. So I'm going to zoom in on the first 2 days because I think that is the critical time of this experiment. And what you'll see is that in either treatment, they lost more weight than the ambient treatment, the control. But in the joint treatment, they lost the most under the joint stressor scenario. So under that scenario, they lost 35% of their fatty acids, whereas the ambient treatment only lost about 20%. So here are broad conclusions from that research. Larvae are the most vulnerable to starvation when under joint stressor scenarios of warming and acidification. And we need to understand food limitation in order to predict climate change impacts accurately on larvae. I'm going to bring us back to our overlying question, what is happening to baby lobsters? I've just highlighted that temperature and acidification may be contributing factors to the restriction of the bottleneck, causing fewer larvae to make it to that post-larval stage. Now I'm going to address another possible driver of this disappearing act, and that is one that's indirectly tied to climate change. Spoiler alert, they all kind of are. So this is a study that we looked at how shifting size of maturity in adult females may be affecting larval performance and resiliency. This is the larval quality question. This is a question is tied to a study that was produced by DMR, Department of Marine Resources, that noted that as temperatures have been warming over the past 50 years, lobsters have been producing eggs at smaller sizes. So on the x-axis, we have carapace length, which is telling us the size of a female adult lobster. On the y-axis, we have the proportion of proportion mature. In other words, what percent of female are at that size are producing eggs. And the different lines here are different year groups. So this graph is, time is moving left here. And it's depicting that over time, females are reaching maturity at smaller sizes. This graph is a little counterintuitive to me, so I remade it for my own brain. Over time, temperature is increasing and lobsters are becoming moms at smaller sizes. So we've seen this pattern, but what we don't really understand is whether or not those small moms are able to produce the same quality larvae as those large mothers. This question was addressed by a recent PhD graduate from Rick Walley's lab, Alex Asher. He went out and he measured eggs and larvae coming from small and large mothers, and also tested quality and resistance to starvation. And he found that smaller mothers have fewer eggs, and those eggs are smaller. He found that smaller mothers also have smaller larvae. And lastly, he found that larvae from those smaller mothers had poorer nutritional status. So they were more vulnerable to starvation than their large mother counterparts. So yes, if we have more larvae of lower quality from small mothers, fewer may be making it through all these stages, which might be contributing to this explanation of the Great Disconnect. And Alex has a pending publication on that. So more information might be coming out soon for that if you're interested. All right, so we've talked about climate change and larval quality. Now I want to talk about the role of food limitation and how it might be playing into this. And I'll try to keep this pretty quick here. This is the most recent body of work that we've done with some new collaborators listed here. We know so little about larval lobsters as compared to the adults. We aren't even really quite sure what they're eating in the wild, let alone if they can find enough of it. So in the lab, they're generalist eaters, and they'll eat almost any type of small crustacean you give to them. Typically, lab experiments have used Artemia as a food source. This is a picture here. Those are sea monkeys that you might have raised as a kid. Artemia aren't a natural food. They're brine shrimp, but they're not great nutritional value for lobsters. I want you to compare that to a picture of a wild-caught post-larval which is out in wild eating a mixture of food. And you can really see the difference in size here. These guys are eating a mixture of different copepods, probably other crustaceans. This is a barnacle nauplii, or a larval barnacle up here. And they might even be eating calanus, which is a really big fatty copepod. That's actually what the right whale is targeting up here. They're really good nutrition. Notice I said could be eating because we aren't actually sure what they target in the wild. We know a varied natural diet is better, but what we don't understand is if there's like a critical food source with nutritional components like calanus, for example, that improve survival of these animals. So as the Gulf changes, the food communities are changing. Who is there and what quantities of who is there might be really important to understand. So that led us down multiple avenues. And before we can see if they're food limited, we have to figure out what they eat. And I'll be highlighting a couple experiments where through field observations, dissection, gut analysis, and feeding experiments. I'll start with field observations. These happened over 3 summers during the season that larvae are out there, June to August. The goal of this project was to find out where they sit in the water column. We don't even know really where larvae are vertically, and what the food communities there might look like. So this is done in 2 parts. To catch the lobster, we dragged a net horizontally at different depths. Niemisto is a term for the very top surface layer, so we dragged it there. We also did it at 10, 20, and 30 meters. And so using this method, we could figure out what stages of lobster were at what, congregated at which depths. We also dropped a zooplankton net, which is a vertical net, and this net was actually able to close, so it's a finer mesh, so it can catch smaller things, and we dragged it from 40 to 20 meters, closed the net, brought it up, dragged it 20 to 10, closed the net, and then 10 to the surface. And so we were able to correlate different communities with different lobster abundances. And finally, we dropped bottles to these depths to collect water for eDNA analysis. Environmental DNA, also known as eDNA, is a process of collecting free-floating strands of DNA from the water and sequencing it all and trying to figure out what kind of organisms might be there. So all the animals that are swimming in the ocean are constantly shedding their DNA into their environment all the time. So you can actually, if you can find those sequences, you can figure out what species are there without pulling any of those organisms out of the water, which is pretty neat. All right, so these are some results of the lobster abundance. These are from the 2022 seasons. We have our depths here along the side, and along the y-axis here, we see the larval density. One thing I want to point out here is that the densities of these individuals were in the single digits per 1,000 cubic meters. That's equivalent to a 1 cubic meter is 1,000 liters. So this is over like 1 million liters of water. So 1 per million liter of water. The point is that they're very sparsely populated. It's much easier to raise them in tanks and from eggs, turns out, than catch them in the wild. So the colors are representing different stages. We have lots of this dark green post larva and light green stage 1. I'll also point out that we found very few 2s and 3s. It's still a mystery as to where they are. We can't find them. But the last thing I'll point out is that the post larvae were mostly up in the Niemisto, zipping around at the surface. At this stage, they're big, they're good swimmers, they're really good eaters, and they're trying to cover distance. So it's typical to find them up in the surface. It's kind of like if you were trying to count all the kids in the community and you went to the school to count them. So stage 1 are a little more spread out, the light green here. They tend to get stuck in the seaweed at the surface, but there was this trend of, as you move down in the water, they increase 5.5 times. So there is some evidence that younger larvae may be distributing themselves deeper, where conditions are more stable and they're a little safer from predators. And guess what? The types of food you find at different depths also change. So copepods and these crustaceans called cladocerans dominate these systems, but they're found mostly in the top 20 meters. At deeper depths, we find a higher concentration of early stage, where we find the lobster larval stage 1. We also find baby copepods, baby barnacles, all these really small, slow-moving organisms that would be really good targets for stage 1 lobster. And this will come into play later. Okay, so now we know what the lobster might be eating based on who we see them associated with. And I'm going to bring it back over to Alex, who looked at what they were actually eating. And what he did was actually, with the lobsters we collected in the field, he would dissect out their stomachs and actually look inside to find clues as to who they were actually eating. So as you can imagine, it was mostly fragments because these organisms tear and chew their food up. But amazingly, he was able to find distinguishing body parts in here. So this is like the tooth of a Centropodes, which is a species of copepod. He was also able to take the stomachs out and process them using eDNA analysis to find fragments of different things in the stomach to ground truth what he was seeing through the scope. And from this, he was able to determine that wild-caught lobsters are generalist feeders. Their gut contents are mostly crustacean prey. And he also corroborated this with his eDNA analysis. So they were eating what's available to them, but what if they have a choice? This is where Evie's work came in. She's a recent master's graduate from Rick Wally's lab, and she was looking at prey preference through feeding experiments. What she did was, she was looking at how effectively all these stages of larvae feed on different types of prey, and also set up these choice experiments where she gave them a choice of different food items and observed what they preferred to eat. And what she found was that stage 1 larvae tended to most effectively eat small, slower prey items that were easier to catch, and that post-larva were super feeders. They could eat more than she could ever put in the jars. And when they had the chance, they would target Calanus finmarchicus. As a reminder, those are the copepods that are the big fatty oceanic copepods that right whales target. And they're unique in producing these wax ester layers that are really good food for many fish and other marine organisms. So all in all, Evie and Alex's findings complemented the field analysis. And we started to see, paint a picture of what these animals are relying on for food, which made us ask, okay, can we see any evidence that they're food limited? This is the last little bit here. We looked at this by analyzing time series, which already exist through other databases. So they help us find relationships between different organisms. I'm stealing this screenshot directly from one of Rick Wally's presentation because it highlights the findings of some work that he did. It utilized data collected from the NOAA trawl surveys on lobster abundance, a Norman Doe plankton survey on larval and zooplankton abundance, and then the ALSI database, which measures the amount of young of year settling lobsters they find at the bottom in any given year. And these are really long time series that have been going on for a long time. What it found was that the eggers in stage 1s were increasing, but that post-larva and new settlers were decreasing. And they compared the year-to-year abundance to many different prey species, and they found the closest relationship to actually be with Calanus. This is a cold water species. It's predicted not to do very well in a warming Gulf, and their decline in numbers may give us evidence to the statement that larvae may be food limited if this is a critical part of their diet. So we wanted to see if we could find any physical evidence of food limitation, which requires our last experiment. Again, this is included in Alex's work. In this experiment, larvae were maintained in tanks and either fed an overabundance of natural zooplankton diet or starved. Using this method, we could create a scale as to what a well-fed larva looked like in terms of lipid profile as opposed to a starved larva. And then we analyzed larvae caught in the field to see where they sat on that scale, and to determine if they're food limited. So here's some quick results of that experiment. This is just one of the fatty acids they looked at. It's an omega-3, which is an important fat for these larvae and is a good indicator of nutritional status. This value is normalized to their overall mass. And we see that the well-fed larvae are up here in the blue, and the starved ones are down here in the green. It's likely they burned through their omega-3 stores while in that starvation mode. So let's see where the field larvae lied on that scale. So they fell well within the scale, having a bit more fatty acids than the starving, but quite a bit lower than the well-fed larvae. And we took this to be evidence that larvae in the field, at least during this time period, were in fact food limited. Okay, so I just showed you a ton of data, projects, names, collaborators, and findings. In terms of the overall question of what's causing this disconnect, I feel like we can take the question marks off of these arrows, because we saw that it's likely that in some way, all of these things are contributing to the decline in the number of post-larvae that we're seeing. Of course, as scientists, these answers only open more question, and there's more things that could be studied that could be contributing to this, such as predators, shell disease, microplastics, marine contaminants. But understanding these dynamics are important if we want to adapt to a changing ocean and mitigate negative effects on our fisheries and ecosystems. All right, with that, I want to say thank you to our funders and everybody I mentioned in all these slides, listed on the bottom of all these slides, amongst others. And thank you for listening, and the Boat Shop for having me.
Wow, Maura, that was amazing. Thank you. And so much excellent information. You also, anyway, I just feel like I learned so much in the last 45 minutes. So thank you for sharing all your excellent knowledge. All you wonderful folks out there, Joe Guten Tag, let's get those questions into the chat box. But I do have a question to start off with, Maura. If fewer larvae are making it to maturity, when should we start to see that reflected in the commercial lobster catch?
So this question is really eloquently answered in Rick's ALSI database work, because it's able to project, based on those young of year lobsters, how long until you see that reflected in the landings. It takes 7 years for larvae to reach the size in which they're being landing. I'm always a little bit careful about making any declarative statements in terms of like when or how much something might be impacting the lobster industry, just because it's so tied into a lot of people's livelihood and identity. So I'm going to refer that to the ALSI work that Rick has done. But the indication is that the uptick isn't going to last forever. They are kind of moving northwards into better water for their metabolic systems.
Why is freshwater increasing acidity?
It's not so much that freshwater increases acidity, it's that it lowers the buffering capacity of seawater. So there's this quality of seawater called alkalinity, and this has been a few years since I had to talk about this, so forgive me if I misname some things, but it determines how much the water can take in those free hydrogen ions that come from the chemical reactions that happen when CO2 dissolves into the water. they're able to kind of buffer against that to a certain degree before you start seeing those drops in pHs. So that like pillow, that buffering capacity actually is degraded by fresh water, and it has to do with the chemical components of that fresh water.
Okay. What can we do? What can be done to slow the warming of the Gulf of Maine?
I mean, I think that this is the Gulf of Maine is warming at a faster rate because the Gulf Stream is shifting, and the Gulf Stream is shifting so that like more of that hot, salty Gulf Stream water is entering the Gulf, as opposed to that cold Labrador water coming up from Canada, that colder water that keeps the Gulf really nice and cold. The temperature of the Gulf is an interplay between those two bodies of water, as well as, you know, how the air temperature is affecting that surface layer. Any kind of like climate mitigating things are helpful. I mean, that's such a, you know, such a hard question to answer nowadays in terms of like what an individual can do, because these things tend to have a rolling process. I mean, I think that the best that we can do is try to be responsible citizens, vote in ways that promote science and climate action in these ways. But the train's moving.
Do lobster fishers tend to stay away from the river outlets?
Because of the buffering capacity?
I believe so.
So a lot of these rivers are very estuarine, and you can actually fish up into them. The acidification, we're not seeing it really affect lobsters right now, especially adult lobsters right now, in a way that like we're overly concerned about. I know some people in the shellfish industries are a little bit more concerned about it. This is really like future projections and trying to understand how it's affecting the larva. But in terms of, I don't think that the fishermen are avoiding the river outlets. I mean, we're out there and we're avoiding pots all the way up towards Wiscasset. So, you know, these rivers are, a lot of them are estuarine and pretty salty, pretty far up.
Sure. Okay. Has this research been discussed with the Maine lobster industry? And if so, what has been their reaction?
So this research has been, like, a lot of it has been in a collaborative effort with the Lobster Research Collaborative, run through Department of Marine Resources. They're very good at like keeping dialogue open with industry. Also, I know that some of this has been presented Rick was running the International Lobster Town Hall meetings for a long time. So this research has been out there. In terms of their reaction, I mean, I think that lobstermen in Maine are concerned about the outcome of the fishery, whether that be like issues in climate change, immediate issues like changes to the Gulf of Maine that are human induced, like you know, in the salmon aquaculture and the wind energy. I mean, the guys are usually pretty plugged in to what's going on, and I think that they're concerned about like keeping the species and the fishery going and in good standing. I will say that there's been some really good work out of GMRI that has highlighted like how Maine fishermen have done a really good job keeping this fishery sustainable with things like notching the female eggers and re-releasing them. Some of these things that they were doing long before they were even mandated here. And as a result, the fishery has lasted in southern Maine, you know, longer than expected if you looked at similar situations in southern New England. So I think that, you know, they're listening. I'm not I don't have a lot of interactions with them myself. I feel like one step removed because I'm in the zooplankton world, but yeah, I don't know if that answered the question, but
I think so. Okay, I have a few more, then we're going to wrap it up. Is there any research being done on the possible impact of salmon farming on lobsters?
not that I know of. That's a really good question. I don't know. All of our salmon lice work has been in Norway, actually. But I know that there is some salmon lice work going on at UMaine, and that's a really good question. I might ask around on that one.
Do schooling fish like bluefish affect the larvae population?
Yeah, so that's a really good question too. That's one I didn't even touch on, which is what are the things that are eating the larva? That's another way that if something new species or like changes in fish species are targeting lobster larvae as a critical food source, that might also be another thing. That's not something that we've looked into at least yet, but it's another avenue of questioning for sure.
Okay, folks, this is your last opportunity to ask questions. Joe?
As the lobsters move with the climate change, and they're moving north, the food for the baby lobsters, I got the impression, and again, I have to admit this is not my field, but that the food for the baby lobsters is not keeping up with the lobsters, which are laying the eggs. So are we going to have more larvae produced as the lobsters go north, and in the future, even less food for the larvae?
It's it's a really good question, and I think it depends on what it is that they're eating. So if it's something like calanus, which is that cold water oceanic, if that is a critical part of their diet, we would see really close ties in the fluctuation of calanus and then the fluctuation of lobster in terms of those early stages. I think that the larvae are generalist eaters, so anything that they can catch, if they can find something to replace those food sources and get those nutritional value, it could be okay. Like there are types of zooplankton that are moving northward into coastal Maine that we haven't really seen here, like past the Cape in the past. And those might be good food sources. I can't think of any that have like such good lipid reserves that are like prime bread and butter for marine organisms. But yeah, I mean, I named some things that we're hypothesizing. We still don't really understand their diets. I feel like we're just starting to poke at it. So I think that's something we're going to pay attention to and that we're all curious about.
Thank you.
You're welcome. Thank you.
Is it accurate to say that the herd is moving north to Canadian waters?
So the trend has been that it's the population dynamics have been moving northward, and the fishery has closed in southern New England or has pretty much petered out, and those conditions are no longer hospitable. So they're moving to deeper water, they're moving northward. Unless it happens at a rate that they have enough time to like adapt as a species, I think that the suggestion is that they're going to be starting to move northwards toward Canada. Although the Bay of Fundy stays pretty cold, maybe I don't know if it'll happen in our lifetime, but that's kind of the suggestion that's out there right now. That I gotta say the adult physiology is kind of where my expertise grows a little fuzzy, but
sure. Thank you. This was a fantastic evening of learning together, and thank you all for being here this evening. And Yeah, this is absolutely fantastic.
Thank you so much. I hope everybody has a nice night. Stay warm.
Yeah. Okay. Bye, everyone. We'll see you soon.